Okay, hello class. This is where we left off in lecture today, and I was looking at the notes for Monday. We have a lot of material to cover on Monday, so I wanted to just run through the last 8 slides or so on video, let you watch this over the weekend, get your notes so we can be caught up and ready to hit the ground running on Monday morning. So we finished by talking about the pathway in the bottom-up signaling process, where the nerves come into the NST and travel up to either the PBN and amygdala for the emotional content of the taste stimulus, or to the primary taste pathway through the VPM of the thalamus up to the primary insular cortex, and then the secondary OFC for flavor. Alright, next we need to talk about neural coding. And I want to be very clear that neural coding is different than neural pathway. Neural pathway is the physical pathway that the signal takes to get from the receptor up to the brain areas. The neural coding is the signal within that pathway that allows the brain to understand what physical stimulus was present. So here we see recordings from a chordae tympani nerve, a peripheral nerve from the anterior part of the tongue going up to the brainstem. And the blue line represents when different stimuli were placed on the tongue. And so you see in the top trace here, we have salt that's on the tongue, and there's not much of a change from baseline firing. Then we see sucrose, we get an increase in firing. We see sour, hydrochloric acid, not much of a change in firing. Quinine, bitter, not much of a change in firing. And at the bottom we have just water, spontaneous activity, that's your baseline activity. So what type of a neural code would be represented in this particular neuron? What does it respond to? That's right, it just responds to sucrose. So that just responds to sucrose, any activity in that nerve means that there's sucrose present. What type of coding is that called? Labeled line. Labeled line. There you go. All right, now a second neuron. Here we have water in between the blue lines, and we have salt, sour, bitter, and sucrose. And what do you notice to the responding patterns for salt, sour, bitter, and sucrose? Do they look different if you just look at the action potentials? So for salt, we get sort of this moderate, continuous stimulus. For sour, we get a very vigorous, intense stimulus. For quinine, it's a very interesting on-off, right here, on-off, 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 biphasic response. And then for sucrose, we really don't get a response at all. So, is there a pattern of the neural signal here that corresponds to different taste qualities? Yeah, so we would call this an across fiber pattern, where it's the pattern of the code that lets the brain know if it's salt, sour, bitter. Okay, so this is an experiment that we did in the laboratory where we were recording from 50 different or 40 different single chordae tympani neurons. And you see that we had some shown here that responded best to sucrose and really didn't respond to salt, sour, or quinine. If we look and organize the other 40 neurons based on which stimulus they responded best to, see that we have some that are salt best and some that are acid best. If we take that and look at this a little bit more detail, here's neuron 1 that I've highlighted in pink on the screen. It responds a lot to sucrose, not very much to salt or sour or bitter. If we look at this neuron, now we have a neuron that responds primarily to salt and not to anything else. So these two neurons would be labeled line neurons, one labeled line for salt, one labeled line for sweet. Neuron number 1 being a sweet labeled line neuron, neuron number 7 being a salt labeled line neuron. Then we have a bunch of other neurons like neuron 15 and neuron 27, that respond broadly across salt, sour, and bitter. And this is another type of neuron that we usually that we can find. And we would call these generalist neurons. And right now we're just looking at the total number of spikes per second. We're not actually looking at the pattern of the spikes. And it turns out that the pattern is important here because the brain's able to discriminate through the cross-fiber pattern between whether or not it's a salt, sour, or bitter. So we end up with some sucrose specialists, or labeled line, some salt specialists, or labeled line, and then we end up with groups of generalists, where we have some that respond best to salt, but also respond to sour and bitter, some that respond generalists to sour, but also respond to salt and bitter, and then we have some bitter generalists that also respond to salt and sour. And again, this graph is just showing the mean number of spikes per second. We have to look with these generalists at the pattern of the activity in order to understand how the brain can recognize the difference between salt or sour or bitter. So there's been a long time debate in the taste community about labeled line versus a cross-final pattern. And the most recent evidence that I just showed you tends to support a role for both, right? And that each type of coding may be used for a particular function. So one theory is that labeled line is important if you simply want to detect a particular stimulus. So what are the two taste stimuli that use labeled line? Sweet and salt. And what's important about sweet and salt? Those are the two nutrients that we can't make in our body that we have to eat in order to stay alive. We can't make salt. We need salt for our electrolyte balance and for neural signaling. We can't make sugar. We need sugar for blood glucose levels. Very important to maybe have dedicated lines that tell us when we have found salt or sweet in a particular food. The cross-fiber pattern is used to discriminate between similar stimuli. So some sour stimuli are good, some sour stimuli are harmful. Some bitter stimuli, like broccoli, are good for us. Other bitter stimuli, like poisons, are bad for us. So using a cross-fiber pattern, we can get much more subtle coding results where we can do better discriminations. So doing better discriminations, not just detecting what a stimulus is, like the sweet and the salt, could lend itself to a cross-fiber coding. Psychophysical studies tend to support this. We have better discrimination across salts and sours and bitters than we do between different sweeteners, for instance. Okay, so that's neural code. Gustatory system uses both labeled line for sweet and salt detection and a cross-fiber pattern to discriminate between salt, sour, and bitter. Now, back to our gustatory CNS areas. There's actually a very sort of complicated circuit, as we see here. The GI is the gustatory cortex. Here you see the thalamus, the VPM, the PBN, the NST at the bottom. And you see that there are things like the amygdala, the lateral hypothalamus, the amygdala over here weighing in, the thalamus over here having a different role. And you see that these arrows are are bidirectional, not just unidirectional, so there's descending influences back into here. So this gets very complicated. I'd like to simplify this by looking at this circuit. And in this circuit, we have the nerves, the three nerves coming into the NST. From the NST, we have three primary outputs: the PBN and amygdala, the hypothalamus, and the primary taste cortex. So the primary taste cortex primary taste pathway goes through the VPN of the thalamus, ends up in the insular cortex, and then the frontal operculum, the OFC, for that flavor perception. We also have reciprocal, and that's pretty much a one-way street, right? Inputs coming in, afferent taste inputs coming in, ending up in our taste perception area. Now, we also have connections from the brainstem for the hypothalamus here. And you notice the hypothalamus with hunger and satiety is bidirectional. We're going to talk more about this on Monday, But that means that activity related to hunger and satiety in the hypothalamus can actually come down here and influence the afferent taste signals that are coming up to primary taste cortex. So whether or not you are really hungry or really full can change the flavor, your flavor perception of food. Likewise, with the PBN amygdala, there's also this bidirectional pathway. So your emotional state, the emotional value of stimuli can influence your perception of that stimulus. And so changes in your emotional state, like a learned taste aversion, can make a food that used to have a perception that was very good, now have a perception that's very bad. All right, there's your review points. We are caught up. Don't forget, reports due Sunday at 8 o'clock. We have lab from 2 to 5. Enjoy the rest of your weekend, and come ready on Monday. We got a big slideshow, big topic to talk about.